as well, and that still seems to be moving steadily.
Yes.
Not changing rate at all.
Thermocouple still steady at 20 to 21 degrees. Thermocouple's above 40, 41, 42 degrees. Thermocouple still quite steady at 21, 22 degrees. GT thermocouples.
Sorry, Sean, would you be able to speak up a little bit? Sorry, I can't hear you.
Oh, okay. I should speak up a little bit?
Yeah,
okay.
just a little bit. Sorry. That's
Yeah, no
great.
problem.
Thank you.
Super thermocouple is 22, 23 degrees, and IGT electronics thermocouples are around 31 degrees. Now super went up to 26, 27 degrees, and IGT is still at 32. Super is now 23, 24 degrees, and the IGT is about 40.
Lee, how are we on time?
Obviously, we're a little bit behind because we had a delayed deployment this morning, but we're doing okay. We're doing okay. I think by the end of our watch, we would have hopefully wrapped up this site.
Okay.
In terms of doing, all your filters, flush your bags, and then sample the bags, collect the rock, deploy the marker. I think it might be deploy the marker, then collect the rock.
Okay.
But yeah, what will be with it. We'll reassess, I think, at the end of this sample location. And
okay. My flow rates, they look lower than we expected. So I'm just wondering if I should, add some additional time, or if that's going to take away from. I'm still getting at least 5 liters on the filter, which is a lot. That's good, but we were hoping for 15.
Okay.
So
I would get a viable sample for yourself. So don't short the time. If you need to extend the time, let's make sure you get enough water from here. Would be my gut to make sure that we get a good viable sample for you. So don't worry about time. You get what you need from this site.
So just confirming, super filter 1 was supposed to get 15 liters, but you think it only got 5?
It got 5, yes.
Okay.
And what's your minimum acceptance?
According
is 5
to
still good, but it's obviously closer to 15 would be ideal.
Yeah. Yeah. We have no idea what the biomass is in this location,
of course,
you know, but we were hoping to get 15 liters.
Yeah.
And I can't, if I go up to the faster speed, it might blow out the filter.
I was going to say, that was my next question. I noted that it was slow speed, and I was like, what would the fast speed do? Obviously, it would put holes in your filter, which then ruins your sample entirely. Okay, so how much longer do you want to extend?
Is it okay if I extend them from 15 to 30 minutes? So that would add 30 minutes to this sampling.
I think so. I think
if not, we can just stick with this, and then if we need to filter more the next dive, we can make that adjustment later.
Super wand temperature is at 17 degrees, and IGT is at 20 degrees.
Because we would have to filter that much at the other site too.
So once you start, once you make the decision to move the flow, to increase the volume or increase the time to 30 minutes, then you have to increase all of the time frames to 30 minutes for each one. Okay. I mean, I don't know the implications for your samples, the viability of the samples. So my gut would be to do what you feel is necessary to get a viable sample from here. Because what I don't want to do is say, no, stick to your 15 minutes and then you don't get what you need out of it. And I know that's a difficult call to make, but I think if you would prefer, then let's go with the...
I think I'd rather stick with the dive plan and just do 15 minutes each filter, and then since we didn't find a lot of diffuse flow sites, we might be coming back here.
Okay.
Oh, well, actually, we are coming back here.
Yeah.
To pick up the samplers. So I'll just stick with that.
Stick with
since
15.
We're already behind, and
okay,
we'll get what we can and keep on keeping on.
No, that sounds good.
All right.
No, that's
Super
fine.
wand is at 17 degrees, and IGT thermocouple's at 33 degrees. So it's dropped off a little bit, but it's still fairly stable. Super temperature's coming back up 20 degrees. To 20 degrees, not up 20 degrees.
So just to check over, you're happy with all of that? You've got that logged, you're on?
Yeah, I think so. Just making sure that we clarify the difference between what should have been the final volume and what is the actual final volume that we collect.
Perfect. So yeah, 5. Great. Thank you.
You're listening to Nautilus Live off the coast of California and Oregon at Gorda Ridge. Subsea scientists are collecting samples to study regarding hydrothermal vents, and presently they're collecting at a particular site.
Hey, Trevor, how are the ground faults for Super and the IGT?
Rock solid, 3.6 meg right now.
Wonderful.
Perfect, thank you.
Yeah, you're welcome.
We're kind of down to around 16 degrees.
IGT temperature is currently at 40.
Okay.
IGT thermocouple. I got to stop calling it IGT because it's not actually a gas tight. It's just the electronics from the gas tight and the thermocouple that we took off one of the gas tights and strapped it to the Super wand for redundancy.
Nav, this is data.
We're about 19 or 20 degrees now.
Well, we got some time. I figured we can get the sampling numbers all set up.
Okay, great.
We have about 5-ish minutes left.
Hold on, let
on
me.
filter 2.
Having trouble hearing you. So am I going to drop multiple targets or just put in the comments as each sample?
So we're going to drop one target, but I'm going to have you name it something really specific.
Okay, because I dropped a target that's NA108-007 for the first.
So if you do NA108-007.
Yep, I have.
And then dash.
Uh-huh.
I'll record for the laptop.
013, and I may have you change that later depending on if we actually get all of those samples.
Okay, say it's.
So yeah, make it a dash instead of a underscore if you can.
I see, and that's the only difference.
Yeah. Yep.
Okay.
And then in the same name, do like dash 007 dash 013.
So
it's sample 007 through 013.
I see.
Yeah.
All
Perfect.
Right.
And
okay,
then
great.
in the notes, there's going to be a different naming scheme.
Okay.
Super thermocouple is reading 17 degrees.
So it'll
and
be
IGT thermocouple is reading
H1751.
38 degrees.
Sorry, 1 second. I'm going to turn off. Say the naming scheme.
H1751
Uh-huh.
underscore, and then parentheses, S1 through S3, comma, S7 dash S8.
Dash S8.
Yep.
So super
And-
sample, okay.
And then parenthesis, underscore, mount with a capital M, Edwards with a capital E.
One word.
Yep, perfect.
All right. Great, thank you. So that's super sample 1 through 3, and then 7 through 8.
Through 8, exactly.
Okay, great. Oh, yeah.
How are we doing on time, Amy? Are we
Just gonna do that.
I have about 2 or 3 minutes left.
just preparing ourselves?
Okay.
Over here, for logging. Perfect, thank you.
Sure.
Super thermocouple's at 22 degrees. IGT thermocouple's at 35, 38 degrees. Super thermocouple's at 16, 17 degrees, relatively stable over this time. And IGT thermocouple's at 27 degrees. Super is still hovering around 16, 17 degrees.
Okay, I'm going to stop filtering.
Okay, confirm that.
Okay. Filter stopped.
Thank you.
Okay, that one was about 4.75 liters.
Do we have a max temp for that one as well?
I think it was 30. Okay. Going to port 3.
Sorry, Amy, was that the start of 3?
I'm going to port 3.
Oh, you're going to
filter
port 3?
Yep.
Okay.
Begin filtering.
Copy that, begin filtering.
Confirmation on the flow meter. Super is hovering around 20 degrees, 22. The IGT is 5174. About 50 degrees. Temperatures still hovering around 19 or 20. $18.
Can we have an update on the temperature discrepancy between the two? Now you're back, Sean, as well.
Yeah, I'm back now.
Perfect, thank you.
It's 20 degrees and stable, super.
IGT temperature is about 51 degrees, climbing to 59. I stepped out to get a power cord for my laptop and heard that my kids can hear me on the Nautilus Live, so that's kind of fun.
Oh, great.
They were pretty excited to hear my voice, so coming across. It's good to know that someone's listening.
They're listening.
We're down to about 15 and 16 degrees again.
IGT thermocouples at 35. Hi, Nathan and Sarah, if you're listening. I'm not sure if we're allowed shout-outs, but I did it anyway.
Do it.
Everybody's got to get in there. Hi, moms.
Yeah, yeah, hi, Mom, because she's probably sitting with them as they're watching this or listening.
I think you're allowed to say hi. I think you won't get in trouble.
Okay, good. They can put me in science jail.
No having fun.
Super thermocouple is 17, 18 degrees, and the IGT thermocouple is at 62 now. I think those temperature readings from the IGT thermocouple are real. I have no reason to believe they're not. It seems to be a little bit more wafty than the super, which is probably closer to the end of the intake, probably a little further down in the fluffy stuff, if you will.
It's incredible just watching it actually to see the variability in the fluid flow, to see how it's...
Yeah, you can actually see it waft back and forth. It's getting temperature agreement, and that is nice too. Super thermocouple is at 27, 26 degrees currently. IGT is about 30, 28. And we're actually pretty close to even right now.
Sean, how will you store the gases that you collect so that you can take them back to the lab and study them over the next few months?
Some of the gases we'll actually measure out here. We'll put a small metering valve in the end of the IGT, and we'll very controlled and carefully let out a small amount of fluid, which should contain the gas appropriate for that fluid. And then we'll put it into a syringe and do what's called a headspace extraction. We'll basically allow the gas that's in the fluid to come out of the fluid into the headspace of the syringe, and then inject it onto an instrument to measure the concentrations of methane and hydrogen. And some fluid we will put in evacuated glass bottles with rubber stoppers, and we'll take those back and analyze them back on shore for CO2 and then isotopes of methane and CO2 most likely.
Do you see a certain type of like methanogens at certain types of smokers, whether they're the clear, white, or the black smokers?
Certainly the biology will impact the isotopic composition of the methane and CO2 if there are microbes that are either using or producing those, but I haven't measured them here, so I'm not really sure.
Mm-hmm. Thank you.
We have about 7 minutes left on this filter.
Well, my shout out and potential for science jail work, the kids heard me say their name, so they were pretty excited. I'll happily go to science jail now. Super thermocouples at 21, 22 degrees, and IGT thermocouples at about 33.
Every time you go to say that super thermocouples thing, I hear the supercalifragilisticexpialidocious in my head.
That's really what it's short for, but we just like to keep things anachronistic in science.
Yeah, gotta stay efficient, right?
Somebody really needs to make a temperature sensor or some kind of instrument with a name like that. An acronym, actually, like that.
Oh, God.
That would be a challenge. I'm not even sure NASA could do that.
That's like half the alphabet.
Super thermocouple is 22, 23 degrees, and the IGT thermocouple is 28 degrees.
Next time you just got to say it as fast as possible.
Yeah, then I'll get tongue-tied, and that might not be pretty. I'm not sure there's an 8-second delay.
As a student interested in the blue covering on that probe, and I believe, but I don't want to be quoted on it, that Eric said he 3D printed that, but I don't know the material offhand. It wasn't the standard filament. Is Eric available?
I believe he's sleeping.
Okay.
He was up all night trying to work on the super.
Oh, right. Okay.
Yeah.
Super thermocouple is 21 degrees, and IGT thermocouple is 32 degrees.
So Lee, when I fill the bags, I might need to go a few minutes over because, and I'll just keep an eye on it because we can put the camera on it so I can see if it's inflating.
Of course,
yeah,
sure.
Because I don't want to half fill them, then we won't be able to get all our experiments done. So
no, absolutely. You get them as full as they need to be. It's not a problem at all. And we have to flush those 3 times, is that correct? To get rid of the DI water and then, okay,
yeah,
no problem.
About 3 minutes left.
Super thermocouple temperature 21, 22. IGT thermocouple is at 45, 48 degrees.
I hope that's stuck in your head every time you say it now.
Yeah, I was just thinking you're
Yes,
going to be saying
it is. Thank you.
I was as well.
I'm
They're
gonna-
right. Yeah, successfully done.
The wee hours of the morning being exhausted, the supercalifragilistic temperature sensor is measuring a spoonful of sugar.
Yes, I like that.
Super thermocouple is, yeah, now that is definitely in my head. Thanks for that. Is 1920 degrees, and IGT is 22 degrees.
We'll just have to turn this into a hydrothermal vent musical in the van.
No bad can come from that. Super temp is now at 17, 18 degrees. And IGT thermocouple is at 40 degrees.
40.
40, yeah. 35 now.
The actual super thermocouple, is it inside the blue intake tube?
Yeah, the way Eric explained it is that the thermocouple is in a separate tube that is part of the blue super wand.
Okay.
So the blue super wand is a support structure for the, I'm assuming it's Teflon, but I'm not certain of that, wand that is actually the super wand. And then the thermocouple is sort of embedded in the super wand going down the side of it.
Actually
so that the tip of it, yeah.
I see, okay. But the tip's exposed. Then.
Yes, again, this isn't my area, but that's just from what.
That's how you understood it.
That's how I understood it from Eric. He can tell me I'm wrong later.
No more than I do, that's why I'm asking. I'm just intrigued.
Well, I can certainly make it up if I don't know it. Super temp is 18, 19, 20 degrees, the last few readings I saw. And the IGT temp is 39. Amy, do you get any sense if the flow rate is slowing down or not?
No, it doesn't appear to be.
Okay.
And the last filter was almost exactly the same as the previous filter in volume.
Hey, Science, can I take bubble?
Can we take the bubble cam to do our gauge check?
Oh, yes, please.
Thanks.
Super temp is 19, 20 degrees. IGT temp is 50.
Okay, I'm stopping the filter. And we're at 19, 20 degrees at the end.
Final IGT temperature was around 51 when I stopped.
Okay, thanks for that. So we can confirm that's stopped.
Just calculating my volume. 5 liters. Okay. So now I'm going to advance to a bag port. I'll advance to port 7 and do bag.
Did we have a max temperature for that last filter?
don't think I remember the high temperature. 20 to 25.
Okay. So just to confirm, so we're going to switch ports now, and
yes.
then do we have to switch our super probes around as well? Is that correct?
That's correct. And then I will need to be able to see the bags inflating as well.
Okay. So if you're ready to do that, I can get Trevor to...
Trevor.
Go ahead.
So I finished the 3 filters, and now I'm going to start the bags, and we have to switch the inlet to the exhaust.
Roger that. Okay, come wide, please, Jess.
And then if I could get the bags on camera.
Yeah, you bet. Okay, so I'm good to remove this and stow it?
The wand?
Yeah.
Yes.
Okay.
Okay, I'm starting the move to port 7.
What do I want to do here? What do I want in life?
Trevor, do you need bubble?
If you're finished with it, yes, please.
I'm not, but I'm struggling with it, so I'll give it to you.
Okay. Can I get preset 5? And then tip down a little bit? Yeah, get off of there. There we go. So I'm going to need to take off and re-land to get this out. See how it's all stuck up in there?
Okay.
So are you good for that now?
Yes.
Okay. Can I get front porch view, just preset 1?
Yes, sir. So we're having to reposition to be able to get access to the wand, is that correct?
I'm just going to pull it out and then re-land
so.
in probably
Okay.
about the same spot. Just kidding, taking off. Whee!
Just checking. We're under strict instructions to go back to the same spot, so I think we've got good visual confirmation of where we need to be, and we'll just... That's great.
Push it in farther to make it harder first, before I pull it out. Oops, that's not the right button. Okay.
For those joining Nautilus Live, we're off at Gorda Ridge by the Juan de Fuca Plate and Pacific Plate. And we are sampling hydrothermal vents. This is off the coast of California and Oregon.
We're going for that same spot, right?
Yes, please. We don't have a temperature probe this time, so if you remember the exact spot, that would be perfect.
This is a test
Okay,
of your
we've been
abilities.
staring at it.
Yeah.
We've been staring at it here on the back grid.
Okay,
so
maybe you can help guide.
Yeah, between the three of us back here, I'm sure
I've
we
been
can.
staring at my computer screen.
Should be just to the left of the middle of the screen. Is that what we're doing?
Let's have some zoom action and see if we can find it. Go ahead. So I'm thinking it's, let me bring the arm there and point at it.
That looks like the same worms. So
Come wide, please. Oh, that's not what I want to do. Why did grip lock come off? That's lame. Yikes.
what is that?
If it helps at all, your previous heading was 54 when you were actually doing the sampling.
Roger. Oh my god. That is springy.
Do you have a good view in bubble?
I do not.
Is that better?
Oh man.
There you go, you're close.
Looks like the exterior of some of those funnels have already been colonized by things.
Yeah, I noticed that earlier. That's really cool.
What's tenuous? Is it gonna spring back on me? Maybe not. Yeah, it is. Oh, okay. Trouble.
Super springy bungee on there.
Super springy bungee.
The springiest of bungees.
Yikes. That is not user friendly. Alright, I'm trying a different approach here. You know what?
Put it back and restart.
Yeah. There's no sense in fighting this for way too long and
I was going to say, you're going to have
losing.
to lift off again. Okay.
That's better.
There you go.
Aubrey was 054.
Yes.
Alright, get front porch again.
Yep. I think it's that group of rooms right in front of you.
Are you good on the bubble?
Almost. Yeah, that looks stable. Yeah, you can take bubble. So we're going for that same spot before. So, can you zoom in, please? Good there. It looks like it was about this area. I'll get it more vertical, but this area is where I'm going for.
What do you
I think so. I think it was a little bit more sort of in the background, so back a tiny bit.
Sure. Okay, come a little bit out, please. Yeah, good there.
And more to the left, you see where that bare rock is? Just, yeah, in between the two there.
As in?
Yeah, about...
I'll get closer, but right about here?
Yeah, but not actually touching the...
Yeah, you're in the
substrate. You need to come up.
How's that look? Is that still too close?
I think in terms of like actual locality as to where the wand end is, that looks in the right place. It's just whether it's in the right place for you, Amy, and it's not going to suck any of the...
Understood. Zoom in again, all the way. Yeah.
Oh yeah, that looks okay.
Looks okay there?
Yeah.
Okay, I can maybe line it up a little better for a little more zoom.
Those are all baby tube worms.
Yeah, you got any more zoom there?
Oh, these ones, yeah, these are the paralvanella.
That's all
So
you got.
I think
Thanks.
this whole mat is a matrix of tube worms and gastropods and biological substrates. So I think it's all just
it's amazing.
A huge mass of biology going on. So hopefully full of microbes as well.
Yes.
Yes.
Got a little bit of everything.
So you locked in, Trev?
Yeah, it looks pretty solid.
Okay.
If you're happy with this point, then I can make sure it stays there.
Okay, yes.
Okay. Great.
So I need to flush the bag 3 times before I start filling, so I'll let you know when I change.
Okay, and if you're not getting a good enough view with camera 7 there with the bags, we can put that on a bigger screen for you too.
Yeah. Where's camera 7?
On the multi-viewer, not the big center bubble one, but up and to the right.
Yeah, could you try a different one?
Make it bigger?
Sorry.
Let's
Yeah.
do it. It's here.
There you go.
Oh yeah, that's much better. Okay. So I'm gonna start with 7, which is the one on the bottom. So could we get that one in view?
That is as in view as it gets.
Okay.
This camera does not move.
It has some pink drawings on it. You can see the pink, so hopefully that will help guide us. Okay. Okay, I'm starting to fill the bag slowly for 30 seconds.
So this is the flushing process? Confirming that this is the flushing process?
Yes. Okay. Now I'm going to purge that for 30 seconds.
So it's 30 seconds of pumping and 30 seconds of purging, is that correct? Is that how this works? In terms of flushing it, you pump for 30, fill it, and then purge for 30?
Yes, that's correct. And I'll do that 3 times. Okay, I'll pause. Filling the bag slowly again. Pause. Purging. Okay, filling last time. I have confirmation that the wheel is moving. Okay, now I'm purging. Stop. Done with washing. Filling bag slowly.
Perfect, thanks Amy. So we can confirm the flushing has been complete and we are now filling the bag.
We are now filling the bag.
So an anticipated rate of 1.3 litres per minute, it says here it should take approximately 5 minutes. So do you think you'll have to extend that time just to make sure you do get a full bag?
I might need to, yeah. I'm hoping that I'll be able to see the bag inflate. It's going to be more difficult for this first one, but...
What is your anticipated final volume for the bags?
They hold up to 4 liters.
And in your little bag quiver that we're looking at in camera 7 at the moment, it's the top one that's filling.
Yeah, I do see the pink moving.
Oh, I see. So it's at the bottom of the screen. Sorry. Yeah, my bad.
Yeah, it's the bottom one.
Yeah. No, I can see it. Oh, so you've color-coded the tops and stuff so you can see when they're inflated.
Yeah.
It makes it a bit
I drew pictures on them.
Nice. Yeah, that's good.
That art is always well appreciated.
It's also possible that the calculations are wrong. There's a K value that Eric put in the spreadsheet, and if it's wrong, then all the calculations will be wrong. So I'll just double check with him again later and make sure that that we have the correct volumes logged.
Oh, okay.
Because it could change if the K value is different. It could change.
Okay. The data loggers would have logged whatever you said at this moment in time, so that can be something that can be altered retrospectively in your files.
Yeah.
Yeah. Okay.
Yeah, just for your
I'll
notes.
just double check with him.
Yeah.
Sure.
I've made notes in the notes and observations section also, but in the sample logging, it's been logged as the anticipated final volume, and then I made a note in the notes and observations under your tag that says what the actual final volume you thought it was at the end of the sampling.
Awesome.
Yeah.
Thanks.
Thanks, Aubrey.
I mean, the bag looks like it's filling, and it looks pretty full from what I can tell.
Yeah, compared to the one in the middle of the quiver where you can see loads of space, it looks pretty...
Yeah. And I definitely don't want to overfill it. I don't want it to pop.
I was going to say, there's going to be a fine line, right?
Then we would have 0 liters. You have 2 minutes left to fill for 5.
Yep.
I feel like it may be enough. It looks like it's pretty full. That looks really full to me.
You call it whenever you're ready.
Okay. Got 30 more seconds. And stop filling.
Copy that. So it'd be interesting to know how your spreadsheet calculates the volume of that. Because it, like you say, it does look full, and at least we know that's a known volume.
Yes. I'm having trouble getting it to stop. It's not stopping. Okay, now it stopped.
Good.
It took me a minute to get it to stop. Okay.
It's okay. Confirm stop.
72.39. Okay, so according to this, it says that it filled at 1.29 liters per minute, and I have 6.43 liters in there, so it's overfilled.
Oh, interesting.
So it looks pretty full.
Yeah.
There is the bags, the tubing to the bags is smaller than the tubing to the filters, so that might have some bearing on it. I don't know.
Yeah, and I suppose, yeah, because I mean, if, yeah, that seems,
Okay.
yeah, because you'd anticipate them to fill at the same rate, but then I suppose, yeah, depending on the diameter of the tubing and then also the amount that could get through your filters, obviously you can only pull so much water through a filter, so that's going to slow down your flow rate potentially.
Yeah.
Would it? Yeah, it would, I suppose, wouldn't it? Okay.
Okay. I'm going to move to filter 8.
Okay, great. So just to confirm.
I mean, bag 8, sorry. Port 8, bag 2.
Just extra numbers. No, it's fine. Port 8, bag 2.
I didn't get much sleep last night.
That's fine.
Okay. Okay, so I'm going to start the flushing of the bag. And start filling now.
Okay, confirm
This should
that.
be the middle bag on our screen.
Okay. Keep an eye on that. Oh, I can see it moving.
Yeah.
And the paddle's moving, although there's a lot of diffuse flow in front of the paddle at the moment. It's difficult to see, but it's definitely moving.
Yeah. You can definitely see it filling up.
Yeah.
Okay, that's paused. Now I'm purging. And paused. Filling. paused, purging.
So this is the final flush?
No, there's one more after this.
One more, sorry.
And pause. Filling. Pause. Purging. I can see the bag deflating. And paused. Now I'm going to fill the bag slowly for 5 minutes, and I'll start.
Copy that. 2036.
So I think on the previous bag it was also counting the flow counts when I was purging, so that might have added to it.
Oh, I see.
Yeah, so
right, hence
it's
the
probably,
reason we've got a bigger volume.
Yeah,
I
it's
see.
probably, I don't know, 3 or 4 liters. I'll see if I can figure it out later.
No, it'd just be good to know, wouldn't it? Just
Yeah,
because if that's a known volume that we're filling,
yeah,
then that gives some kind of guidance in terms for your filters.
yes.